Hi, I'm Brian Coyle. I'm a postdoc in Mike Braun's lab in the Department of Vertebrate Zoology here at the museum, and I'm working as the coordinator for the Red Siskin Initiative, which is an endangered species conservation program that I'm going to talk to you about today. So the red siskin is a small, non-migratory finch that had been common across a broad region of northern Venezuela into Colombia and the islands of Trinidad and Tobago. Today, however, it's very rare and probably entirely extirpated from the eastern half of this range. The largest known population, with an estimated 1,000 to 3,000 birds, was discovered in 2000 in remote southern Guyana during a joint expedition between University of Kansas and the National Museum of Natural History, including Mike Braun, Chris Malensky, and Brian Schmidt from the Birds Division. There were also populations on Cuba, at least into the early 20th century, and there may still be birds on Puerto Rico, which we hope to explore in the coming year. So the main reason that the red siskin is endangered is wildlife trafficking for the pet trade. And this has been going on since at least the early 19th century, maybe even centuries earlier than that. And it remains the #1 problem today. But it increased dramatically in the early 20th century. So German aviculturalists at that time popularized the production of a reddish-colored canary based on hybridization with male red siskins. And as the popularity of this trend increased, the demand for red siskins, for wild male red siskins, also skyrocketed and led to severe overharvesting. So in response to the need for conservation, the Red Siskin Initiative was formed to protect and to restore self-sustaining populations of this bird across its historic range. International partners are from Guyana, Venezuela, United States, and other places, including diversity of private and public institutions, NGOs, communities, grassroots organizations, and many others. And at Smithsonian, there's scientists and experts from 4 biology units who are all providing multidisciplinary expertise and leadership for every facet of the project. It's also an exemplar project of the Smithsonian Institute for Biodiversity Genomics, and it's received generous support from the Grand Challenges Consortia. So the core conservation strategy is primarily to reduce the threat of wildlife trafficking, especially in Venezuela, and to gain a better understanding of the species through research that's going to enable a captive breeding program for reintroduction. So the field research is a major component, and that's focused on characterizing habitat and ecology of the species, as well as testing and developing survey protocols for monitoring over time. And we're also using historical records of the red siskin to develop a habitat model to help understand habitat requirements and also to identify different locations for reintroduction. The molecular component of this project has multiple parts. First, we're going to assess the genetic structure and viability of remaining wild populations and develop molecular tools for managing diversity of captive flocks and for monitoring the populations over time as well. Second, we'll develop a genomic screening assay to make sure that all the birds that are included in captive breeding in Venezuela programs are going to be pure red siskins and not have DNA from other species. There's some cryptic hybrids that look like red siskins, but they have ancestral hybridization, so we don't want to introduce those kind of birds into the wild populations. And underpinning this entire effort is a Smithsonian Biodiversity Genomics program. So right now, the whole genome of a red siskin is in the process of being sequenced at Duke University, and we should have the raw data back very soon for our bioinformatics team to assemble what will be one of the highest quality bird genomes to date. This will fully enable the level of detailed work that we are planning to do. The captive breeding program involves 2 breeding centers in Venezuela and the Smithsonian Conservation Biology Institute. We're establishing a research colony of red siskins at Front Royal, Virginia, under the leadership of Paul Marinari and Warren Lynch. They're going to test and develop methods specifically for siskin husbandry, care, breeding management, and as well as developing techniques for adapting birds from captive environment for release into the wild. And these birds that will be used in Virginia won't be actually released into the wild. What we're going to do is develop a program and then implement that program at 2 zoos in Venezuela, where they will be trained and supervised by Smithsonian. The birds there will be confiscated from traffickers, and they'll be used for the founder of the population. The reintroduction strategy centers around the innovative Smithsonian Migratory Bird Center's bird-friendly coffee. So This program, in coordination with local communities and natural parks in Venezuela, we plan to release birds into shade coffee farms that are specifically managed for red siskin conservation. And this program benefits farmers through better marketing and retail opportunities, but also recruits farmers, surrounding communities, and coffee consumers in support of the conservation effort. And lastly, and this just shows a red siskin actually in a coffee farm, so we know that they use them by choice. So this is what inspired our thought to use the Migratory Bird Center program as well. And lastly, I just want to highlight that this bird is, it's like a poster child for conservation diversity in Venezuela. It's on the highest denomination currency, the 100 Bolivares note. It's on the cover of the Red Book of Threatened Species and considered one of the top priority species for conservation in that country. And there was a Smithsonian-led conservation workshop in 2014 that made the front page of one of the biggest city newspapers. It was also in news, media, internet, TV interviews, and radio. And so it just shows the close kind of societal attachment to this bird, and it could be leveraged not only for the red siskin, but also to promote conservation of other species throughout the region. And lastly, I'd just like to acknowledge many of the members here at Smithsonian and our institutional partners that have helped contribute to this project.
This question is for Brian. I'm interested in the confiscated birds from the pet trade. With the genome, are you going to be able to pinpoint their geographic origin so that you're not mixing up the gene pools in the wild?
Yeah, well, that's what we hope to do. We have geo-referenced museum specimens from across Venezuela, and we've collected live birds as well from different locations, collected blood samples from birds in different locations over the last few years, and so we are surveying, to the extent possible, the genetic diversity across the whole country, and we'll do the same thing in Guyana. So, but yeah, we should be able to have a pretty good idea, and we intend to be able to try to pinpoint where the birds are from.